There we go. So I'm a PhD student from Duke University, and today I'm going to be telling you about using a single-cell approach to understanding sexual development in Plasmodium falciparum parasites. So the reason we're using single-cell is because the Plasmodium falciparum life cycle is very complex. And so most of the parasites stay within the asexual part of the life cycle, approximately 95%. But then you have a few parasites that commit to a sexual fate, which is called the gametocyte form. And so these gametocytes form in a 3-to-1 ratio of females to males. And then these gametocytes are develop over 2 weeks into the mature stage 5 form, which is the only stage that is transmissible from the human host to the mosquito vector, where mating takes place. And so in terms of Plasmodium, let me go back for a second. So today I'm going to actually cover 2 different projects. I'm going to cover a single-cell qPCR using the sexual stages, and then I'm also going to cover a project using single-cell mRNA-seq on the complex part of the life cycle. And so in terms of transmission, only the sexual form is transmissible from the human host to the mosquito, so this makes sex a necessary and obligate life cycle step. And the majority of antimalarial drugs actually target asexual stages, and many are less effective against gametocytes. And recently, there's been a study showing that male and female gametocytes actually exhibit different responses to antimalarial drugs. And so in terms of Plasmodium falciparum gametocyte expression, there are over 300 genes that are upregulated during sexual development. And a few of these have been revealed to be sex-specific at later stages, but there's very little resolved in terms of earlier development. And so previously, sex-specific reporter lines have been used to flow sort populations and determine male and female sex-specific expression. And so this is a figure from the Cell paper in 2005 showing that male and female gene expression is actually very distinct. And you can see here that there are even a number of proteins that are not known for their functions in male and female development. But using fluorescent reporter lines can often be biased. And sometimes these markers are debated. And so this is why we're taking a single-cell approach to understand the process of gametocytogenesis in males and females. And so when you're using bulk cell populations, you have a whole bunch of cells here, and you're going to have a few that commit to a gametocyte fate. And a few of them are going to be male, and a few will be female. And then furthermore, in terms of their sexual development, in the end you get a mix of male and female gametocytes. And so when you do any bulk cell analyses on these cells, you're only getting an average gene expression across all cells, and you cannot reveal any sorts of rare cell types. And additionally, this means that only a single-cell approach can accurately dissect male and female gametocyte development from commitment. And so what we have used here is the C1 microfluidic approach. And so we can capture single parasites using microfluidics. And then in this first part of my talk, I'm going to talk about profiling known gametocyte genes for their sex-specific expression. And so when you look at this figure here, what we first do is we need to isolate the single gametocytes from uninfected red blood cells. So we might Percol purify and magnetically sort those gametocytes. In this project, we used mid-stage and late-stage gametocytes. And then we can load and capture those onto the Fluidigm C1 into this microfluidic chip. And you can see here that there's a single parasite isolated within this microfluidic chamber. And then from there, you can lyse the cell, perform reverse transcription, preamplification with the primers you're using for the qPCR, and then you can harvest your cDNA and either do, for the cDNA, you can do sequencing, or you can do qPCR, which I will talk about now. And so we used 87 gametocyte-specific genes and then 4 controls in order to determine the male and female differences between those genes. And so using 90 single cells, you can see here, this is a Pearson correlation of the genes that we used in our study. And so, this axis and this axis are both the genes, and you can see that in this cluster here, we used a couple known that were functionally studied male and female markers. And so a bunch of genes actually cluster here that are female specific. And then down here we have genes including HAP2 and MAPK2 that cluster as male specific. But what was interesting to us was there were a bunch of genes that clustered in these areas that were not previously shown to be male or female specific. And so when we look at the single cells themselves, you can see that there are a small number of males, and they cluster very separately from the females, which are down here. Additionally, the females separated in a stage-specific manner, so our cells that were collected on day 5 clustered separately from the female cells clustered on day 9. And so when we look at this figure here, this heat map, we can see that this is the gender-specific gene expression that separates male and female single cells. So on our y-axis over here, we have our panel of gametocyte-specific genes that we used in our study. And then on the bottom here, we have our single cells. And this includes two positive and negative controls. And so what is very distinct about this heat map is that you can see a very small number of males that cluster separately, and they have very distinct gene expression. And so when we looked closer at the genes that were being expressed, we saw a number of markers that were already known to be male-specific, but then we also saw a very strong upregulation of genes that have not been described or functionally studied for their male specificity. And then furthermore, you can see where the male expression is, there is a downregulation of the genes that are female-specific. And in this female-specific cluster here, there are a number of genes, again, that were previously not uncovered to be female-specific. Furthermore, as in the previous figure, the stage 3 and stage 4 or 5 females cluster separately. So you can see an upregulation of gene expression here. Another interesting point is we actually use merozoite-specific protein 1 as an asexual control, and you can see that there's an upregulation of this gene in a few cells, suggesting the heterogeneity of the population and potentially a functional role of this gene in gametocytes, but that would need to be further studied. And so this is these are dot plots of the individual gene expression of the genes that we used for male and female specificity. So these are genes that are enriched in males. And so what you can see here is that our two top candidate genes have not been previously described for their male functions, and they are very highly male specific. And then what I also want to point out is there is this gene here. This is actually previously shown to be an early gametocyte gene, but in our data set, it's clustering as male-specific. And you can see other genes down here that have been shown previously for their male-specific functions. And we also see the same correlation in females. What I want to point out in this plot to you is that we've studied 8 AP2 factors in our study, and this is an AP2 factor here, and it actually clusters as female-specific, which is very interesting. And so this will need to be studied for its biological role. So in terms of the first part of my project, some conclusions that we can draw are that male gametocytes are distinguished from female gametocytes by their transcriptional profile at the single cell level. And then these mature females actually exhibit heterogeneous expression and appears to be a stage-specific increase in expression. And the implications for this are both drug development. As I mentioned, male and female gametocytes show different responses to drugs. And I'm really interested in determining how these genes could be involved in mating type and gametocyte development. So the next part of my project I want to talk to you about is the development of mRNA, single-cell mRNA-seq for Plasmodium falciparum. And so we captured single trophozoites, and we wanted to compare those to what we know is different. So we used 5 single late-stage gametocytes as a comparative control. And again, you can see that we can capture these parasites here. And so actually, the CAFSAC lab last week published a paper in Nature. They used DROP-seq to understand sexual commitment at the single cell level, and they found epigenetic regulators involved. But here we're using the fluidyme C1. So in terms of what we see is you can see that on the bulk cell level, you see many more genes that are expressed. And then when you look at the single cells, the gametocytes show higher transcript expression than the asexual cells. And they're showing, instead of showing above 4,000 transcripts expressed, they're showing between 1,000 to 3,000. So we took these single cells and we did PCA clustering. And so very nicely you can see that the gametocytes cluster separately from the asexual cells. And then what we see 3 distinct populations of asexual parasites. But first I'm going to talk about the gametocytes. And so when we do a cell-to-cell correlation, these are the cells and these are the single cells. You can see again very nicely the gametocytes cluster very strongly together in terms of their gene expression. And then each single asexual cluster also clusters very strongly with one another. So just focusing on the gametocyte cluster here, we saw that there were 340 markers upregulated in our data set in the gametocyte population. And so what I did is this heat map over here is showing the top 100 genes. And so I took 10 genes that were not previously described to be gametocyte specific, and I validated those using bulk cell qPCR in stage 5 gametocytes. And so you can see very nicely that there are a number of genes that are more highly upregulated in the gametocyte population. This is uncovered at the single cell level. So taking this a step further, we wanted to remove the gametocytes from our data set in order to see how is the asexual population how are those cells different from one another? And so what we're seeing here is that in cluster 1, we see an upregulation of the Sera genes. In cluster 2, it seems to be exported proteins. And then cluster 3 is rope trees. So seeing the Sera genes and the rope trees separately does suggest that this could be a stage cycle-specific imperfect synchronization. But what's interesting is that even these cells, they seem to correlate pretty strongly with one another, but there is heterogeneity within the population. And so what I'm trying to do now is to try and understand how is each cluster, these cells in each cluster, how are they different from one another? And so in terms of the conclusions and future directions from this study, we use single-cell mRNA-seq to uncover heterogeneity that is masked in population analyses. New genes are expressed during gametocytogenesis. They've been able to be identified using this method. And what I am currently working on is RNA fish validation to determine either are those clusters time-specific or is there cellular heterogeneity. And I can take that a step further within each population to determine the cellular heterogeneity of gene expression. So with that, I would like to acknowledge my lab. I'm in Ashley Cheese Lab at Duke University. In terms of the computational analysis, the Duke GCB core with David and Helene have been very helpful. I need to acknowledge my funding source, the NSF, and this work has also been funded by the Burroughs Wellcome Fund. So with that, I will take any questions.
So should I have time for a couple of questions? Any from the audience? Yep.
I just wanted to make a comment. One of the things you might want to check, especially with respect to the male-specific genes, this is something that we noticed when we did some gene expression profiling, is there's a lot of uncharacterized genes during gametocytogenesis. But if you look for their orthologs in other species, including in the human, you'll often find that they're expressed specifically in sperm cells.
That's actually, so the second gene that I show in that dot plot is actually called meiosis-specific nuclear protein 1, and it's necessary in using knockout mice. It's necessary for sperm flagella formation.
Yeah, so there's very nice gene expression for that you can use for that correlation. Very nice work.
Thank you.
Anyone else from the audience? Okay. I have sort of one. You mentioned on the first slide the sort of 3-to-1 ratio that's been found between sort of female and male.
Yeah.
And just sort of wondering that one of the plots, it seemed like that was slightly off from that ratio. So is that sort of well defined as a ratio, or is it sometimes lab dependent or field dependent?
It can be lab dependent, field dependent, strain dependent. So I think in my case it could be partly strain dependent. I use strain 3D7A in these experiments, and that's been shown to be about 12% male specific. There's also been a study showing that if you have a high parasitemia, that could also increase the male to female ratio. So I think those could be playing roles in maybe not having as many male cells as I would have liked.
Thank you very much, Kate.
Thank you.